Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, Spatial Tissue Proteomics to Assess Health and Disease. Please join me in welcoming Dr. Fabian Coscia, a renowned expert in spatial proteomics and your speaker for today. Dr. Coscia is the group leader at the Max Delbruck Center for Molecular Medicine in Berlin, where he leads groundbreaking research at the intersection of high content imaging, deep learning, and ultrasensitive mass spectrometry. So, with his extensive experience in developing and applying single cell and spatial tissue proteomics workflows, Dr. Coscia is a pioneer in the field of deep visual proteomics. It is our great pleasure to have Dr. Coscia share his insights with us today. So following the presentation, we will also have a Q&A session and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And without further ado, I will now hand over to Dr. Coscia to begin his presentation.
Yeah, thank you very much. So I will first, of course, thank the organizers for the invitation and for setting this up, such a great online event. I think this is really a fantastic opportunity to not only share knowledge, but also to foster international collaborative research. So the title of this meeting here, this online meeting, is Advances in Proteomics and Metabolomics. And in my talk specifically, I would like to focus on advances in clinical proteomics, more specifically in spatial tissue proteomics that you see illustrated here. And so my group in Berlin at the Max Delbruck Center focuses on developing and applying spatial tissue proteomics methods, particularly in cancer systems biology, on the basis of ultrasensitive mass spectrometry. But let's maybe first start with some very general background why mass spectrometry-based proteomics, or short MS-based proteomics, is such a powerful and important tool, not just for basic research, but in general also for translational research. Mass spectrometry-based proteomics really summarizes a core technology that comes in very different flavors and subdisciplines, really allowing us to look at various aspects of the proteome, important for basic science, but also for clinical research. For example, you can take a look at, you can analyze and study protein abundance in cells, for instance, in the tissue context. We can study post-translation modifications, so signaling networks that are happening in basically every second in our cells. We can look at protein turnover measurements to really see which proteins are generated, which are degraded. We can also use these techniques to look at proteins in time and space, to explore the extracellular matrix, for instance, and also very important readouts to understand diseases at the global protein level. And then in the last 5 or 6 years or so, we have seen major advances in the sensitivity of our mass spectrometry-based workflows. Now, for the very first time, allowing us to look at cell-to-cell heterogeneity, here really quantifying a few thousand proteins per individual cell. So this is super exciting, and my lab is in particular interested in really leveraging these highly sensitive methods to better understand how cells are organized in the tissue environment, and how cell interactions, how distinct cell neighborhoods shape the disease-related proteome. So spatial tissue proteomics on the basis of mass spectrometry is a very relatively new concept, but has very great potential for translational research in the upcoming years. So what is so different about conventional shotgun proteomics mass spectrometry on the basis of liquid chromatography mass spectrometry compared to low input or single cell proteomics applications? So while these two different workflows are conceptually very similar, what you also see here illustrated in this figure, special adjustments are really needed in order to deliver these trace sample amounts, so basically the proteome of a single individual cells or from very few cells in the tissue microenvironment, to our mass spectrometer. And really the community has developed over the last couple of years different sample preparation workflows to really allow us to, after protein extraction from a cell, we can then really isolate the proteins. We can use in a very efficient manner enzymes, so trypsin, typically used to generate protein fragments, and then separate these small amounts of sample on a liquid chromatography system, and then use ultra-sensitive mass spectrometer in order to measure as deep as we can and as quantitative as we can the global proteome of these cells. So this is on the one hand a big leap forward in terms of how we process the samples, really using sub-microliter volumes and robotic workflows. But also there have been a lot of innovations from the industry side. So the vendors that produce these instruments to really deliver highly sensitive and innovative hardware that can really deal with these very low, relatively low input ions that come from our biological sample. So these developments have also greatly benefited the field of spatial tissue proteomics, where the ultimate goal is to really understand how cells are spatially organized in the tissue, and how the disease-related proteome is influenced by the cell neighborhood. And just a few years ago, maybe let's say 5 to 10 years ago, bulk proteomic approaches that you see highlighted here in this yellow box have been the absolute standard approach in the field. But today, more and more labs, on the basis of these new sample preparation workflows I just mentioned, and highly sensitive mass specs, we can now include spatial information. So we have now the opportunity to also analyze deep proteomes from very few cells in the tissue microenvironment. And so on that slide, I just want to highlight that this is showing our own work, but this has really been driven forward as a community effort in the last few years, where different groups, very international, on a very international level, developed new solutions that allowed us to really go from the spike level type of data down to really single cell resolution. And so to illustrate a bit these advances in the spatial tissue proteomics fields, I want to shortly talk about one bulk tissue proteomics study that we published a couple of years ago, and then slowly move towards these newer and more innovative and spatial resolved workflows to get an idea really where the field is coming from and what is currently happening, and also then give an outlook in the end where we might be going with such new spatial tissue proteomics workflows. So this was a study together with the lab of Ernst Lengyel at the University of Chicago, where we looked at or asked the question in a very aggressive cancer type, high-grade serous ovarian cancer, if we find proteomic differences in patients that show long-term survival compared to the majority of patients which has a very poor outcome following standard carboplatin-based chemotherapy. So interestingly, there is a small number of patients, around 10 to 15%, that show long-term survival. So meaning after one round of surgery and chemotherapy, the tumor basically is gone and will never come back. So typically we see here long-term survival beyond 10 years without any recurrence. And so this is interesting, as also these women present with highly metastatic stage 3 or 4 cancer. Indicating that there might be some predisposing features in these tumors that makes them extremely sensitive towards therapy and also protect them from any recurrence. Genetic studies could not really reveal or illuminate what is the secret of these long-term survivors, indicating that there might be some non-genetic effects really driving this long-term survival phenotype. So in this study, we applied bulk proteomics applied to archival samples that were taken from the primary surgery of the patients, and in total analyzed 26 tumors using our back in the days deepest workflow we could use in order to analyze around 9,000 proteins per single sample. And so what you basically see here is two different patient groups. So one chemo-resistant group, which showed very frequent and fast chemo resistance and tumor recurrence, and compared the proteomes of this group to a highly sensitive group, including these exceptional responders that did not show any tumor recurrence. Our deep proteomic data, which in total comprised around 9,000 proteins that we could relatively compare between the patient group, did in fact identify a single protein that was highly upregulated in the women that showed long-term survival, that showed chemosensitivity. This protein was at that time largely uncharacterized, a cancer testis antigen 45A. And we could also show that, I mean, this is also just showing the fold change of these two different patient groups, really, that there is a single outlier. And we could then show with our mechanistic work and using a whole portfolio of different mass spectrometry-based proteomics tool, that this protein is not only a prognostic marker of the disease, but it's also a functional driver of chemosensitivity in ovarian cancer cells. And we could lastly also show an important role of the immune system in potentially providing or securing long-term survival in these patients. So while this study, I think, clearly showed that we can identify important new biomarkers from archival patient material, I think one highlight was also to mention that mass spectrometry-based proteomics tools could really contextualize these new discoveries and provide important additional information to really get to the cellular function of the protein and thereby have more biological and mechanistic insights into novel discoveries. So the question is, why could we actually identify the protein using these bulk-based approaches? And so if you look here in the immunohistochemistry picture, so this is an antibody that recognizes this biomarker protein CD45 in the tissue, you clearly see that the majority of tumor cells are really strongly positive for the protein, whereas if you look at this poor outcome patient, there is literally zero signal that you can retrieve from. So this clearly shows, I mean, if you have features, if you have protein signatures that are really shared by the majority of cells, of the diseased cells, you can clearly use bulk proteomics approaches for the discovery. However, in reality, and a large fraction of the proteome looks very different and more like this picture here. So where in fact you see not only spatial heterogeneity within the tumor compartment, where in fact you have regions that are strongly positive, whereas other regions are rather negative, but in the same slide, you can also hopefully appreciate that in different compartments, so for instance, the stromal compartment, you might also have positivity. So bulk approaches would in that case not really help you to really disentangle where the protein signal is coming from. And so here, this is certainly a great limitation where we need newer and more spatially resolved approaches. So I think this slide also nicely highlights that we clearly need imaging-based approaches in order to really get spatially resolved proteome information. And so imaging-based spatial proteomics approaches have been applied very broadly and over many years. And I think they're still very powerful tools to analyze individual proteins or a panel of maybe 30, 40 proteins with very high spatial resolution. So if you plot here their resolution compared to the analytes, you will find imaging-based approaches typically on the side with very high spatial resolution, but with very limited number of analytes of proteins you can study. So this is, I think, quite important to highlight, because if we're really interested in addressing the whole proteome, ideally of a diseased cell or of a diseased tissue, and we're rather talking here about maybe 10,000, 12,000 unique proteins that are being built at a given time, which is of course complemented also by protein isoforms and post-translational modification. So a sheer complexity that we're really not able to capture using imaging-based approaches. So this is where really the other extreme is quite important, and I already mentioned how LC-MS-based proteomics can help us in biomarker discovery. But here the problem is these methods have a relatively low throughput. So while imaging-based approaches can typically analyze 50,000, 100,000 cells in relatively short time, for discovery mode deep proteomic LC-MS-based proteomics data, and we are rather talking about maybe 50 measurements a day or so. So these two highly complementary approaches are of course extremely interesting to combine. with one another to really leverage both of the advantages of these methods. I will also just want to highlight for completeness, there's also other mass spectrometry-based methods such as MALDI-based, so mass spectrometry imaging using MALDI approaches that are maybe right in between these two approaches that also give you a relatively decent spatial resolution, but they are currently very limited in proteome coverage. And throughout this presentation, I'm exclusively talking about LCMS-based approaches that are really the state-of-the-art method for analyzing deep and quantitative proteomes in cells and tissues. So here I think the most important question is how can we really make the best out of these two different approaches? So really combining imaging with based approaches on single or subcellular resolution with ultrasensitive LCMS-based approaches to have a really unbiased, untargeted description of the disease-related proteome. So compared to MALDI-based approaches, LCMS-based approaches don't preserve spatial resolution as such. So we need to really couple these methods with other microscopy techniques, such as laser microdissection proteomics, that then allow us to isolate individual regions of interest or even individual single cells in the native tissue environment context. And then after isolation, we can then extract the proteins and measure the deep proteome. So laser microdissection is an absolutely important and integral part of LCMS-based spatial tissue proteomics, and I will also come to that in a few slides. So now I want to move on, so to say, in the evolution of tissue proteomics workflow and show you the next level, some advancements that allowed us to, for the first time, do cell type resolved tissue proteomics with high spatial resolution using a new workflow that we published in 2022, a pipeline that we called Deep Visual Proteomics. This is a new and maybe first framework that allows us to really integrate imaging-based spatial proteomics with deep LCMS-based spatial tissue proteomics. So the idea is you start with a sample of interest. So this is typically a block, paraffin, formalin fixed and paraffin embedded tissue block from a patient sample where we section the tissue. So we slice it and then transfer it onto a slide for microscopy, where we can then apply either immunohistochemistry, so staining, IHC stainings, or immunofluorescence staining, in order to, first of all, identify all the cells of the tissue. Deep learning-based algorithms are then applied to segment the individual cells, and we can then read out the phenotypic information, so all the image information from all the individual cells in order to classify cells based on their morphology, based on the intensity of the markers that we've stained, to then find morphologically, phenotypically coherent cells that can then be isolated using laser microdissection for a deep proteomic investigation to really then identify what is the molecular and functional state of these phenotypically unique and distinct cell types or states. One very important aspect of the Deep Visual Proteomics pipeline is laser microdissection. I already mentioned it on 2 slides before, where we have now worked on a new workflow that allows us to, in a more streamlined fashion, transfer the cellular coordinates of the cells of interest that come right out of the image analysis and then pass them onto the laser microscope, where then one by one the cells of interest are isolated, and we can then deeply profile their proteomes using the LCMS-based sample preparation workflow. Just to show you one proof of concept study that we did for this publication, we used here human fallopian tube tissue and stain for 2 markers using immunofluorescence staining. So this is a very standard workflow. EpCAM, a cell adhesion molecule we use to highlight, to basically identify the cell boundaries of these fallopian tube epithelial cell, and FOXJ1 is a lineage marker for only one cell type of this monoepithelial cell layer, which marks ciliated cells of the fallopian tube. We can then apply our machine learning-based cell classification, and now cells that are positive or negative will be classified as unique groups. And then here on the right side, you see the individual segmentation masks that then the laser microscope will use in order to isolate these cells in situ for our proteomic investigation. Now, in this picture, you see indeed how this was done. Here we see these contours that the laser microscope sees and then follows for cutting. And here is after cutting, you see how these individual tiny little cells in the tissue were isolated, and then by gravity they are collected into a 384 well plate, where we can then in this inspection mode make sure that in fact the samples were collected for proteomic analysis. Now, moving to the mass spectrometry data, so to the deep proteomics data, we were able to almost quantify 5,000 proteins, unique proteins from these samples comprising roughly 100 to 200 cells of the same type, and with excellent quantitative reproducibility, which you see here, where we see for our group replicates Pearson correlation values for the entire proteome of around 0.96, 0.97, which really indicates that we have a highly robust and sensitive platform here to analyze cell type resolved proteomes. And then this data is clearly showing differences between the two cell types, where in fact FOXJ1, which we used to really gate, to classify these cells, was only quantified in the FOXJ1 positive population of cells, so these ciliated cells, and was completely absent in the negative one as expected. And on top of that, we could highlight many more cell type specific markers in this big data set of more than 5,000 proteins. So this is a great resource to really also not only look at cell type markers, but also to understand functionally what is really the cell doing, what is the molecular function of these, or the tissue-specific function of these ciliated cells. Just one slide on the versatility of the platform. We could also show that the Deep Visual Proteomics pipeline can be used in combination with cell culture, with preclinical models. So for instance, in this example, we could also show that not only can we isolate individual whole cells using our segmentation algorithms, but we can also do the same for nuclei, so subcellular fraction of the proteome. And so these different collection modes are then also reflected in our quantitative data, where we clearly see proteins related to transcriptional and nuclear processes enriched in our nuclei samples, whereas in the whole cell samples, in fact, you see functions related to cytosolic pathways and so on. Now, so the Deep Visual Proteomics pipeline that was published in 22 was kind of the very first landmark study, for the first time really showing how we can bridge the gap between imaging-based approaches and LC-MS-based proteomics approaches. And from there on, many things has happened in the last 2 years. So my previous group, so the group of Matthias Mann, they also developed a newer method that really allows us to now go down to single cell resolution. And in addition, we have a number of technical advances and further improvements to further streamline the entire sample prepper workflow, to make our entire workflow more sensitive. That means we can work with less material. And also there is quite a number of biomedical use cases already on the way, where we try to really as best as we can leverage this new discovery proteomics pipeline and to better understand diseases at the global protein level. Now, for the last part of my presentation, excuse me, I will also talk a bit about our work on single cell tissue proteomics, where we basically further improve the workflow to really get true single cell sensitivity in the context of an FFPE and archival biobank sample. And so this is a study we published end of 23, where the idea was to optimize all the necessary steps in the tissue proteomics workflow, not only optimize really high sensitivity mass spectrometry acquisition and sample preparation, but really starting one step earlier, how we can optimize the staining procedure in order to then, in the end, still being able to capture and quantify whole proteomes, or as deep as we can, let's say, proteomes from few or even individual cells. So the work that we did was mostly focusing here on liver tissue FFPE samples that allows us to really very consistently analyze samples over serial tissue sections. And so this tissue type is mostly comprised of hepatocytes. So we can that way also be a little bit more certain that what we measure here is due to distinct profiles of hepatocytes that make up around 60 to 70% of the liver cell mass. We isolated then an increasing number of cells or small tissue regions that you can see here, and went down from really down to one cell even. So this is in total 300 square micrometer of the region, up to regions that comprised roughly 50 hepatocytes. And what you can see using this optimized workflow, we did the first showing that from single hepatocyte slices, we can quantify around 1,500 proteins and up to close to 5,000 proteins from 50 cell region. Coefficient of variation analysis that you see in this panel here also showed that our protein readout was highly reproducible, with coefficients of variation of around 20% in median for the single cell samples, and around 10% for the higher loading samples with 50 to 100 cells. So this is a plot showing you also how the mass spec raw data looks like using the Bruker-Timstorff SCP instrument, where from a single cell to 12 cells to 100 cells, you see a very strong linear increase of the mass spectrometry signal coming really from only one cell or from roughly 100 times the signal, clearly showing that our method is able to really capture these extremely tiny amounts from single cells. We also looked at the pathway coverage of our proteomics finding and compared the single cell proteome measurements to the 50 cell measurements, and were quite excited to see that for many biological pathways, in particular for metabolism associated pathways like fatty acid metabolism, TCA cycle, and so forth, we had very comparable coverage, so number of proteins we could identify per pathways. And in fact, this is not shown here, but in a similar comparison, we also compared our data to very deep 10,000 protein deep data set, resource kind of data sets in the literature, where in fact we did not find many more proteins for many pathways, clearly showing that using these single cell based assays, we can already compare or learn a lot about the metabolic profile of these cells, and really for the first time look at cell to cell heterogeneity in the tissue context. And in fact, my previous group, they also looked more specifically at the spatially resolved proteome differences in mouse murine hepatocytes, and could clearly show that in liver there's a very strong association between the spatial location of the hepatocytes and the proteome of these cells, indicating really that we, using these highly sensitive mass spec methods, we can now for the first time really address and analyze the cell type and spatial resolved proteome of individual cells. So in our paper, we also showed how we can now use this pipeline in combination with whole slide immunofluorescence imaging that is shown here in the example of human tonsil tissue, where we optimize our workflow to stain 3 to 4 markers in this section, thereby outlining different spatial niches of B cells in germinal centers, and also T cell regions and epithelial regions. Then guided by the immunofluorescence image, we can then classify the cells based on their cell type, based on their spatial location, and then quantify with these cell type specific changes or unique proteins that for instance, you see here the CD3 receptor characteristic for interfollicular T cells. We see B cell receptor proteins only quantified in the B cell zones. And in fact, ikaterin, a marker for epithelial cells, was highly upregulated in the squamous cell epithelium, nicely showcasing the robustness and specificity of our proteomics data. I already mentioned in the beginning that LC-MS-based spatial tissue proteomics approaches currently are very limited in sample throughput. So currently, we can analyze maybe 30 to around 100 samples per day. And to tackle that, we were also interested to basically further work on sample processing to have more streamlined and very automated workflows in hand, where we can then in the future maybe not analyze only 50 samples per day, but maybe hundreds of samples in order to really sample the entire tissue ideally. So here we teamed up with a company, Selenion, that produces a sample preparation robot system called the Selen-1, which is used in the field in particular for single-cell proteomics application that are based on cell sorting techniques. So here, the idea was that we could also directly collect tissue samples into the chip that is used for the Selen-1 robotic, and then in an automated fashion prepare the samples, extract the proteins, digest to peptides, and then have a mass spec ready peptide plate that we can then directly transfer to the mass spectrometer for analysis. So we designed a specific adapter so that we could combine laser microdissection with automated sample processing in the Selen-1 robot. This is showing a little movie how the system now is dispensing a buffer into these chips. And we could also show that using this workflow, we get highly reproducible and robust results. When we compare here our tissue replicates from the same mouse liver tissue of around 50 cell region, we get a proteome correlation of 0.99, really indicating the robustness of this workflow. We also performed a benchmarking experiment to really show that we can also capture cell type specific proteomes using this pipeline, and also analyzed here different cell type specific niches in human tonsils, so B cell zones, T cell zones, and epithelial cell zones. And in fact, we could confirm very striking differences between these two different cell types, and also show that within group variation was much lower than basically comparing all the samples measured here, really indicating that we capture cell type specific proteomes. Now, for this final outlook, I want to just shortly mention where we are heading towards. I showed you some data on the Deep Visual Proteomics pipeline, which is really a new framework and very versatile pipeline that allows us now to connect this spatial proteome readout to other modalities. And there's many more things we will do and want to do in the future. For instance, multiplex immunofluorescence imaging, so spatial proteomics concept is a very attractive next way to combine this discovery proteomics pipeline too, because here we can really look at the tumor microenvironment. We can include staining of different cell types of the tissue microenvironment, like T cells, B cells, macrophages. We can look at stromal markers and then really use all these phenotypic information to then classify ideally all the cell types of the tissue microenvironment and then select the cells of interest. At the same time, we can also couple this concept to metabolomics. I'm sure there will be very exciting talks in this session later on, where we can, for instance, take a serial section and then use reference points to identify exactly the same tissue regions from a consecutive section, and in that way, not only analyze the metabolic profile, but also the proteomic profile for then multimodal analysis. So multi-imaging is an exciting avenue in this direction. And also we have already teamed up with labs focusing on spatial transcriptomics, where in a very similar fashion we can combine the RNA readout with the protein readout to get a better understanding of the disease-related alterations. So I think to maybe sum up, I think we currently live and work in extremely fascinating and exciting times with many innovative technologies out there, in particular in the spatial omics world. And just as we speak, I'm sure there's new techniques that are developed and that will be published very soon. And in particular, together with machine learning approaches and large language model, I think this is a very exciting topic in the future to really leverage these multimodal data and thereby really allowing us to, in a better way, identify novel drug targets, identify more specific new biomarkers, but also gain really unprecedented insights into the health and disease states, and in general, phenotype human diseases at the molecular level. So with that, I would like to summarize my presentation. I hope I could show you that really MS-based, so mass spectrometry-based proteomics is a very versatile and powerful toolbox, not only for basic research, but also in particular for translational systems biology. We recently co-developed a new pipeline called Deep Visual Proteomics, which combines digital pathology workflows, so mostly imaging-based workflows, deep learning, and single-cell type proteomics to deeply phenotype human diseases. In addition, cell type and spatial resolved proteomics data can really illuminate important disease-related mechanisms. So not only to find biomarker candidates, but also protein-based drug targets. So around 90% of all drug targets are proteins. So that's why really proteomic readouts and of quantitative nature are extremely important to nominate, to prioritize these new candidates. And then I think it's quite exciting on the basis of single-cell proteomics technology, so ultra-sensitive mass spectrometry. We will for sure see more innovations in that area also when applied to archival biobank samples, to tissues. And currently, we could already show that from a sensitivity point of view, it's already possible today, but their sample throughput is still a major challenge, which is, I think, why in the future the field will also further develop more streamlined, more robotic, automated workflows to also push the boundaries of that aspect. And then finally, low input tissue proteomics is, I think, a very interesting and a modular pipeline that could also well integrate with other omics approaches. So I already mentioned a few, such as spatial transcriptomics, where we, I think, in the end need to acknowledge that every technique, every method has its own weaknesses and benefits. So here really, I think the most important question in the upcoming years will be how can we best leverage these unique pipelines to really take the best of all these different technologies and of the different concepts that are out there. With that, I would like to thank my group, in particular my fantastic team. I want to, of course, thank all our collaborators, all the support over the last years. In particular, I also want to mention my previous lab, the lab of Matthias Mann, my former colleague Andreas Mund, with whom I developed the Deep Visual Proteomics pipeline, and also Peter Hobart's lab, also my long-term collaborator, Ernst Lengier, and all our clinical partners here in Berlin, and also collaborators at our Max Delbrück Center, and in particular, also here the funding sources are very important to mention. And you, I would like to thank for your attention, and I'm very happy now to take any questions that you might have. Thank you very much.
Thank you very much, Dr. Coscia, for your fascinating presentation. Before we dive into the Q&A, I just want to take a quick moment to remind our viewers, if you would like to ask Dr. Coscia a question, please do submit them to your chat box on the right now. So with that out of the way, we'll start, I suppose, with the big question. Where do you see the biggest innovations coming from in the upcoming years?
Yeah, thank you very much for the question. I think, I mean, if we look back the last 3-4 years, we have really seen remarkable advances and improvements in mass spectrometry, in particular when we speak about the sensitivity. So really, how many cells, how much tissue do you actually need to for a deep proteome measurements? But also throughput has rapidly improved in last years, and currently there is really no reason why these developments should stop within the next few years. So certainly we will have even more sensitive mass spectrometers, more sensitive hardware, allowing us to really dig even deeper into the proteome. We will see higher throughput methods. Currently, we have a throughput of around 100 samples we can measure per day, but it's likely that we also reach hundreds and maybe a few thousands of samples in relatively soon time. The additional topic is, of course, how we really process the samples. How do we deliver them to our machines? So here we also will likely see more improvements in sample preparation. Robotics is really a huge topic, automation. And then in the end, machine learning-based tools to really make sense out of all this very complex biological data, right? So we need new models to really leverage these protein-centered data.
That sounds incredibly promising. Could you elaborate on how AI is playing in a role in these advancements?
Yeah, I mean, so there's many different aspects of AI. I mean, probably the most obvious one is so when we generate these very complex data sets, where you have many thousands of protein sequences that you identify, that you then map back to the protein information. So this is an enormous a task to really make sense out of these very complex mass spectra that you acquire using mass spectrometry. And deep learning algorithms have really completely revolutionized the way we can analyze proteomes to date. So without a deep learning-based algorithm, it would be very challenging to really identify the majority of proteins in our raw data in the end. So this is one aspect of it. Certainly, there is an important topic on data integration, how we can really combine RNA-based readouts, transcriptomic data with proteomics data. Also here, machine learning methods are extremely powerful and important. So, I mean, just in a nutshell, I think there's probably no subtopic that will not be affected by AI or deep learning, machine learning tools in the future. So they're already a big aspect of today's work, and they will even become more important in the near future.
Yeah, and speaking of technologies, how can spatial proteomics be integrated with other omics technologies, for example, genomics and transcriptomics?
I mean, so one important aspect, I think, is in particular if the idea is to really use these omics data to really guide clinical decision making in the future, right? I mean, this is, I guess, one of the very big hopes that these technologies can provide one day. So here, if we have very powerful genomics and transcriptomics methods, I think it's also very important to really validate, confirm that your drug target that you're basically suggesting for therapy is also really present in the tissue. So here, I think proteomics methods can really be helpful to further prioritize which therapeutic strategy might be an option. But of course, also, I mean, for many tissue types or cell types, we also see a disconnect between the RNA and the protein, and only the integration will really show you if the RNA measurement is really a good proxy for the protein, right? So in the end, the more data we have, the more multimodal we can go. I think the better for our basic understanding of the underlying disease. But also, I think you just have more data to really make a clear case and then also guide, hopefully in the future, the therapeutic decision making.
Thank you. That sounds great. So shifting gears slightly, why do you primarily work with archival FFPE samples? Wouldn't frozen samples provide higher quality data?
Yeah, you're raising a very important aspect, and so many people in the field have done this in the past. You comparing frozen tissue samples to archival samples, and indeed, frozen samples are typically of better quality. When we measure a proteome, we can analyze basically more proteins, get a higher coverage. But this is not the most widely the most accessible type of bioarchival specimen, right? So if you look out there in histopathology labs, really formalin fixed and paraffin embedded materials are within the majority of samples, right? So this is extremely important if you want to study many diseases. So you have a very large number of samples in all over the world, right? And at the same time, that we know from, in particular, proteomics experiments that the proteome is very much preserved in FFPE material compared to many, compared, for instance, to the RNA level. So this is important because you can then leverage these very long archive samples that typically sometimes 10, 20 years or even longer in the histopathology labs, and then really leverage all the clinical data that is typically associated with them and really guide basically the way you design the study, right? So you can look at patient outcomes, so you would exactly know up 20 years later, how did the patient respond to the therapy, and then really use this to design your specific biomedical research study. So in a nutshell, yes, qualitatively they are less good compared to fresh frozen, but we know the proteome is preserved, and using these FFPE samples, we can really, in a very powerful manner, design retrospective studies to look at different disease outcomes and trajectories, and really connecting the clinical data with the molecular protein-centered data.
That's a great point. We do have a question, a follow-up question to that previous question. How do you overcome the challenges associated with FFPE samples, such as cross-linking and degradation?
Yeah, so typically what we do is actually very similar to immunohistochemical stains, where heating steps are applied as a very first step before the antibody is administered. And that the reason here is that these formalin crosslinks can be reversed using heat-induced antigen retrieval. So what we do typically when we process our samples for a proteomic measurement, for a mass spectrometry measurement, is that we heat them for typically like 30 to 60 minutes at 90 degrees Celsius, and that helps us to really resolve most of these cross-links in the tissue, and thereby really improving, enhancing our measurement in the end. So there's ways to deal with that, but I agree that it's not exactly the same as as when you would analyze a frozen tissue section without any cross-linking.
Thank you, Dr. Coscia. As we wrap up, I'd like to ask about the future challenges and opportunities in the field of spatial proteomics. So what is the current throughput of your spatial proteomics methods? For example, how many samples can you currently measure per day, and how do you envision scaling up in the future?
Yeah, I already alluded a little bit to it. So, I mean, the bottleneck in the mass spectrometry-based spatial proteomics field is certainly right now the throughput. So we can measure around 30 to 100 samples per day. So one sample could either be a single cell, it could also be a small region of many cells, but that actually remains the same time. And that's, I think, where really the promise is to combine these lower throughput but deep discovery mode mass spec methods with imaging-based approaches, where you can really screen 100,000 cells easily in no time. And so here the strategy would be that you first use a high throughput method. You basically phenotype all the cells, you know the distinct cell types and cell neighbors. You can link them to the clinical outcome, for instance, if a patient responded to therapy or not. And then that way, prioritize the cells you want to then deeply characterize using these low throughput methods. So I think same for RNA and protein correlations and multimodal spatial proteotranscriptomics method. I think here the power is really in combining, leveraging advantages of many different technologies, right? So there is no single, I also mentioned this in the previous talk, there is no single technology that really has all the advantages, right? We always need to find good strategies to really integrate and combine them. And this could be really imaging-based approaches together with LCMS.
Thank you, Dr. Coscia. Just going back into spatial proteomics, I know we've talked about it quite a lot with the technologies and the upcoming technologies. So our question from our viewer here is actually, could spatial proteomics be also used to identify new drug targets or biomarkers?
Yeah, absolutely. I mean, this is really the, in particular, so of course, there's two different ways of spatial proteomics. So imaging-based, you have to pre-select which protein you want to look at, right? So if you do an imaging experiment, you have an antibody that recognizes a specific protein in the tissue, and then you would look at this specific protein. So you have already knowledge beforehand, before you start the experiment. But this is exactly the beauty of combining these imaging approaches with a mass spectrometry-based approach. So this is in untargeted, unbiased approach where we can actually find new biology, new proteins that we really haven't been thinking of, right? So that we couldn't have known beforehand. And this is exactly what you will need if you want to find new biomarkers, if you want to find novel protein-based drug targets. You need this discovery mode approach where you can really analyze and compare thousands of proteins simultaneously and then really only look at the ones that show you where the strongest response, that are most reproducible, and that can also then hopefully be validated in additional data sets. So certainly, I think this is one major advantage of mass spectrometry-based spatial proteomics.
Thank you so much, Dr. Coscia. Unfortunately, that is all the time that we have. We appreciate you taking the time to share your expertise with us today. So any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Coscia, and we'll be back soon with the next talk.
My pleasure. Thank you.